Puji.
Assalamualaikum.
Waalaikumsalam. Ini nanti mulai itu jam berapa? Apa? Ini Rania mana ya?
Jam 9 tepat. Ada Rania.
Oh itu Rania.
Sudah hadir dokter.
Ya.
Apa jam 9 sekarang belum dimulai?
Loh, ini jam berapa? 4.3.
Skedulnya loh jam 8.15 sudah mulai harus di.
Loh, bukan.
Lagu-lagu kebangsaan, lagu dangdut belum ditampilkan.
Dangdut. Mas yang joget jempolnya tok.
Oh,
Rania itu rambutnya diikat ya Rania.
Iya dokter.
Diikat aja. Nah, ikat aja kok kita wobok-woboknya.
Joget oleh dia anduk.
Nah, udah cantik gitu. Ah. Ya. Itu kalau nggak usah pakai masker mana? Ada orang kan di situ.
Oh, nggak apa-apa sih dokter.
Ya, wes. Nah, diikat rapi rambutnya. Lah, wes cantik.
Rania suruh pakai background.
Oh iya, pakai background udah punya belum?
Iya dokter, setelah ini saya pindah ke tengah dulu pakai background.
Wah, cantik kalau gini.
Nah iya.
Gitu rambutnya.
Itu virtual backgroundnya udah punya?
Sudah ada dokter, tinggal dipakai saja.
Ya.
Ya pakaiin sekarang aja virtual backgroundnya.
Sebentar. Nah,
wes sip.
That it was my sound, and that
Nah, ini kan cantik kamu.
to me they
goreng
have a unique sound, and that's
ini sarapannya belum ada ini.
is
Ada gedang goreng di
Kita kok tak transfer.
sama kopi pahit itu sudah disiapkan.
Yang sudah hadir itu 1.800 ini. Yang hadir 1.800 atau 1.900?
Yang hadir iki piro iki?
1.900 ada.
Oh, Astu ternyata ada acara pembekalan, waktunya sama.
Gak apa-apa sih, penting berjalan dengan lancar.
Wah, nggak boleh
Mana
sambil
Prof Lucia
minum.
nih kok? Prof Lucia kemana Prof Lucia ini?
Prof Lucia, Tanya Sedo, masih bekerja.
Wes Rif, nang lagu-lagu Rif. Lah iki lho Prof Lucia.
Mana Prof Lucia?
Itu.
Wah, mantap. Backgroundnya belum diganti itu. Prof Lucia, backgroundnya belum diganti loh.
Prof Makoto belum masuk ya?
Belum.
Bingung dari Jepang. Loh, Prof Yunus ke putih kabeh.
Ya memang. Udah kakek-kakek.
Wis profesor kok. Wis profesor,
tambah
wis
putih
profesor.
maksudnya.
Selamat pagi.
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 Apa kabar, Prof?
Baik. Mudah-mudahan nggak putus-putus ya nanti.
Prof ikut berduka cita ya, Prof.
Kenapa kok putus-putus?
Enggak, sinyalku jelek.
Itu di rumah toh?
Masih
Iya.
di Jogja.
Masih.
Loh, Prof Lia di Jogja ini?
Loh,
Iya.
itu keseripahan. Adik
Oh
iparnya.
iya ya. Iya saya juga.
Iya.
Prof Yunus lupa
iya.
ternyata.
Berduka cita ya, Dek Wanti.
Berduka
Pagi
cita.
Prof Nunu.
Makasih, terima kasih
semuanya.
Pagi.
Pagi Bu.
Pagi.
Apakah sudah masuk Prof Makotonya? Belum ya?
Belum.
Belum ada kayaknya ya.
Universitas Airlangga, kau selalu di dada. Hadirkan lulusan profesional berwawasan luas. Ayo, ayo jadikan Abraham semakin hebat. Melangkah ke depan
Lagunya apa? Lagu saya ya?
Veteriner dan teknologi, inovasi kesehatan hewan peternakan sejahterakan masyarakat. Universitas Airlangga, kau selalu di depan. Junjung etika moral, jaga lingkungan lestari. Ayo, ayo jadikan Abraham semakin hebat. Sarjanamu, doktermu, siap membangun negeri. Inovasi kesehatan hewan peternakan sejahterakan masyarakat, sejahterakan masyarakat. Universitas Airlangga, kau selalu di dada. Hadirkan lulusan profesional berwawasan luas. Ayo, ayo jadikan Abraham semakin hebat. Melangkah perjuangan Indonesia dan veteriner dan teknologi, inovasi kesehatan hewan peternakan sejahterakan masyarakat. Universitas Airlangga, kau selalu di depan. Junjung etika moral, jaga lingkungan lestari. Ayo, ayo jadikan Abraham semakin hebat. Sarjanamu, doktermu, siap membangun negeri. Dan teknologi, inovasi kesehatan hewan peternakan sejahterakan masyarakat, sejahterakan masyarakat. Universitas Airlangga, kau selalu di dada. Hadirkan lulusan profesional berwawasan luas. Ayo, ayo jadikan Abraham semakin hebat. etika moral, jaga lingkungan lestari. Ayo, ayo jadikan NKH semakin jelas. Sarjanamu, dokter hewanmu, siap membangun negeri. Tekuni, teliti, ilmu veteriner dan teknologi, inovasi kesehatan hewan peternakan, sejahterakan masyarakat, sejahterakan masyarakat.
Prof Makoto.
Yes, how are you? Thank you very much.
How are you?
I'm fine, thank you. How are you?
Ohayo
Nice to
gozaimasu,
meet
sensei.
you again.
Gozaimasu.
久しぶりね。
久しぶりですね。
元気ですか、先生。
元気です。
All right. Assalamualaikum warahmatullahi wabarakatuh. Shalom. Om Swastiastu.
Waalaikumsalam warahmatullahi.
Shalom. Om Swastiastu. Namo Buddhaya. Salam kebajikan. Good morning to the honorable lecturers of Parasitology Division from Faculty of Veterinary Medicine of Erlangga University and our beloved participants. My name is Rania Leticia Azara Guritno, an undergraduate student and an assistant lecturer of the Parasitology Division from the Faculty of Veterinary Medicine in Erlangga University as the master of the ceremony in today's event. Thank you for the participants for being here with us today. We are very pleased to be able to welcome all of you to our guest lecture with the topic Molecular Technologies and Basic Approaches for the exact identification of gastrointestinal parasites in livestock, including parasite purification, DNA extraction, PCRs, and sequencing by Professor Makoto Matsubayashi, DPM, Ph.D. from Osaka Prefecture University, Japan. Ladies and gentlemen, before we begin the guest lecture, let us pray to God who gave us the grace to gather in this event. We can pray according to our respective faiths and belief. Prayer begin. All right, thank you. Thank you for the prayers. And before continuing to the next agenda, let us sing the national anthem of Indonesia, Indonesia Raya, followed by Hymn Erlangga. All participants, please remain mute and maintain the proper position. For the next agenda, we have an opening speech by the PIC adjunct professor, Professor Dr. Numudia Retnolastuti, DVM, Master of Science, as the representative of Erlangga University. For Professor Numu, the time is yours.
Thank you very much for Dr. Rania. Good morning and best wishes for all of us. On behalf of Veterinary Parasitology Department, Faculty of Veterinary Medicine, Universitas Airlangga, we would like to thank all participants for attending today's guest lecture, which will be delivered by Prof. Makoto Matsubayashi from Osaka Metropolitan University, Japan. We express our gratitude, especially Prof. Makoto can attend today to give a lecture to students, undergraduate, master, and doctoral program, and all participants. We apologize the Dean and Vice Dean cannot attend this event because they have other assignment. This program is a collaboration between Universitas Airlangga and Osaka Metropolitan University, Japan since 2018. And I'm sorry, Prof. Makoto, and we will renew to memorandum of agreement with change name from Osaka Prefecture University to Osaka Metropolitan University. Maybe next year, 2023, our dean will be done in Japan. We hope that general lecture of parasitology on molecular technology for the identification of gastrointestinal parasites in livestock can improve science and technology, especially for development of parasitology. By giving thanks to God Almighty, I hereby officially open the guest lectures today. Once again, thank you very much for coming and good luck. Amen.
Thank you, Professor Nunu, for your opening speech. And before moving to the main event, we will have a virtual photo session together. So for all participants, please turn on your cameras. All right, so I will start the countdown for the first slide in 3, 2, 1. Moving on to the next slide. And for the second slide, 3, 2, 1. For the third slide, 3, 2, 1. And for the fourth slide, 3, 2, 1. For the fifth slide, 3, 2, 1. For the sixth slide, 3, 2, 1. For the seventh slide, 3, 2, 1. And for the last slide, 3, 2, 1. Once again, thank you for all of the participants. And ladies and gentlemen, it is the time to start the agenda that we have all waiting for. The lecture by Professor Makoto Matsubayashi, DVM, Ph.D. This lecture will be moderated by Professor Muhammad Yunus, DVM, Master of Health, Ph.D., as the lecturer from Parasitology Division of the Faculty of Veterinary Medicine, Erlangga University. Participants are expected to remain conducive by turning off the microphone and listening carefully to the speaker. For the ones who would like to ask some questions, you may ask the question in the Zoom chat, and the question will be answered after the presentation in the question and answer session. Before this event is taken over by Professor Yunus, let us greet him. Hello, Professor Yunus.
Hello.
Yeah,
Yeah.
how are you?
Yes. Okay.
Are you? How are you? Are you fine?
Okay.
Okay, all right. So, Professor Yunus, the time is yours.
Thank you, Miss of Ceremony, MC, Dr. Rania. So, good morning, everybody. How are you today? I think today nice
And thank you.
programs, and thank you very much for special coming for Professor Makoto Matsubayashi. Ohayou gozaimasu, sensei. Okay, yes. For attention time, we meet again in the program and forum to listen together and sharing knowledge for by Professor Makoto Matsubayashi, DVM, Ph.D. from Osaka Metropolitan University. Nice. The new name of after the United People, Osaka City University and Osaka Prefecture University. So today, everybody, ladies and gentlemen, we have the good time to learn and also to listen together with the guest lecture from Professor Makoto Matsubayashi, PhD, DVM PhD, and absolutely he is the incredible lecturer and also some the experiences. So before that, just I will read about his CV. Professor Makoto Matsubayashi, he was born in Osaka, Japan, 23 October 1973. So he is the graduate from the Osaka Prefecture University, and so many experiences he has, and in incredible works and also research and publication. Yeah, I cannot say anymore because so much experiences. So, and then from that experiences, please we listen together and learn more from his competence, because incredible some the experiences, international and national experiences in research and publication. So from the long time ago, from 2003 until now, is so many publication he has. So today is the good time and opportunity for us to learn and then listen together to the guest lecture programs, so that from this event or programs we can increase our and improve our knowledge, particularly his competence in the molecular and parasite, particularly intestinal parasite. And today lecture, the topic is molecular technology and basic approach for the exact identification of gastrointestinal parasite in livestock, including parasite purification, DNA extraction, and PCR and sequencing. So, Professor Makoto Matsubayashi, so the time is yours, please.
Yes.
Okay.
Okay.
Okay. Thank you. Thank you.
Thank you very much. Okay. Can you see? Can you see the my slide?
Yes.
Okay. Okay. Thank you very much, Dr. Nunuk and Yunus and all of the staffs for inviting me into the opportunity to have a lecture. So I am so happy to see you and to have a lecture today. So I will talk about here. The title is about molecular technologies and the basic approaches for the exact identification of gastrointestinal parasite in livestock, including parasite purification and DNA extraction, PCRs, sequencing. So before starting my slides, so first I'm sorry, my English. Okay, sorry.
Okay.
Okay, so I'm sorry. My English is not so good, so I will talk my presentation as slowly as possible. If
Yeah.
you have any questions and if you cannot understand my talking, please ask me later by email or please ask to your professors. So I will start my topics now. Okay. Okay. As Professor Dr. Yunus kindly introduced my profile. So I will shortly explain my brief profile. So I was born at October 23, 1973. I am now 49 years old, at most 50, half a century. So my family is, I have one wife and one son and one daughter. So my hobbies are fishing, movie viewing, driving, and microscopic observations. And this is very important, visiting Indonesia. So
Yeah.
now I often watch Indonesia in YouTube.
Yeah.
I'm visiting Indonesia only in YouTube. So maybe I met the student. I met some students previously, but others can be the first time to see me. So I introduce shortly in here. So about education background, Professor Dr. Yunus already introduced. So I will do it here. So this is the map of Japan here. We have 4 large islands: Hokkaido and main island here, and Shikoku and Kyushu. So as you know, Tokyo is located here. So, and our city, Osaka, is located here. Yes, and this is the picture of Osaka. We have huge castle and huge buildings. Osaka is the second largest city in Japan. Of course, the first largest city is Tokyo. Then, this is the Kansai International Airport at Osaka. You can come to Japan at the airport here. So Osaka Metropolitan University is located here. Osaka Metropolitan University is close to this international airport. You can easily visit our university. Only one station, maybe 5 minutes. So we can easily go abroad and we can invite easily from abroad. So this is my career in the past. but I can delete here. So, but I not here. So in 2011, I stayed in Suban as a JICA parasitology expert. I visited Kota Bandung here. So this is the first time to visit Indonesia. I stayed for a while, for several months in Suban. Then after 2016 to present, we conduct the project about protozoa disease with Dr. April. Maybe he attended today and collaborated with UNEA, Universitat Airlangga, with Dr. Nunuk and Lucia at Surabaya. So finally, so far I have been to Indonesia 16 times. But maybe I will visit again Indonesia more and more and more.
Yeah.
So this is just the introduction or example to visit, to stay at Suban in 2011. This is this is me, a little young. So she was Dr. Sylvia. She was graduated from UNEA also. So I set up the small institute at Suban with them. During my stay, I surveyed some parasitology parasites in the field like this and like this. So we could show some result of intestinal parasite in livestock during my staying. Next slide. Okay. So my sincere feeling to Indonesia, this slide showing. So I'm loving warm and kind personalities of Indonesians, of course. So and additionally, so nice food and hardworking students and so on. So that is why I'm always getting fat. Even during my short stay, I ate so much food. I ate nasi goreng and mie goreng and so nice food and so on. So finally, I have 3 agus and 4 deer as my friends. So many agus and deer. Okay. So instead of visiting Indonesia, actually, many professors kindly visited Osaka. So Dr. Professor Lucia and Nunu and many professors visited our university. So we are so happy and so pleased that they could visit our university. So we are waiting for your next visiting again. So from this slide, I will my topic move to the research explanations. So slide shows today's topics. First, I will talk about general information of intestinal parasites in livestock and introduction of their diagnosis. I heard the attendance will be undergraduate students. So, of course, they need some basic information. So I will talk about basis of diagnosis in livestock parasites first. And second, basic lecture of molecular analysis for the intestinal parasite here. And third topics is about our research cases of molecular analysis. Not published. This data is very, very new findings. So this is just basic information of swine parasites. Red color can be detected by fecal examinations. So as you know, Metastrongylas, we can many parasites in swine. For example, Metastrongylas species or Ascarisum and so on found in liver. In small intestines, we can detect Ascarisum, Strongyloides suis, Eimeria, Cystospora suis, Cryptosporidium, Entamoeba, and so on. Of course, we can find Trichuris suis and so on, were found in ileum. Maybe most of students can explain like the parasite here. For example, in bovine parasites, there are so many parasites here. So, of course, we must diagnose these parasites as a veterinarian in the field. So we must know what's the characteristic of these parasites, of course. So we can see Osteogeria or Trichostrongylus and so on in Abumosam. But in small intestine, there are so many parasites. As we know, or maybe so many species. Eimeria, tapeworm, Cryptosporidium, Giardia, Nematogyrus, and so on. In poultry parasites, we can see many parasites, of course. In Seca, we can see Eimeria species, Histomonas, Meliagridis, Heterachis, Gallinarab, and so on. For example, in small intestine, Eimeria, Ascaridiae, Galli, Capillaria, tapeworm, and so on. So, among these parasites, can you diagnose these parasites using feces as a veterinarian? Of course, you must examine the infection exactly using only small volume of feces. Of course, we can collect huge volume of feces, but in most cases, we just collect small amount of feces, especially in chickens and so on. That is why you must understand how to collect the feces first and how to examine the parasites, methodology, and how to identify the parasites. And of course, you must understand the situation of the animals. Of course, farmers or veterinarians added some drugs or examined animals can show clinical symptom or not, or how to breed these animals and so on. We must record these informations. But you have to know our limited ability at the same time. First, how to collect the feces? You must take fresh samples as much as possible without any contamination. Very important. And recording the information of the animals. So you can use anything for collecting of feces. For example, you can small plastic bag, but you must seal the bag. Or you can use also tubes, for example, of 50 milliliter, if you can seal the feces. Of course, if you use the tube, you must tightly read. Sometimes you can pick up drops of feces, but you had to better take care of contaminations from other hosts. For example, the farmer keep many animals. That is why the contamination will be occurred frequently. That is why we had to better collect the feces directly, one by one. But if you cannot do collect the feces one by one, you can pick up the dropped feces. But please be careful the contamination and the parasite, especially nematodes from the soils. So this is a very, very simple examination method we call direct smear examinations. This is sometimes important for trophozoite. I will explain why. You must note the viability of the parasite. This is very important. For example, you can see sometimes trophozoite. For example, trophozoite of entamoeba, Gerardia, Toritocomonas, and so on. These trophozoites must be examined as soon as possible because trophozoite can be dead soon. That's why if you want to check the trophozoite of this parasite, you pick up small amount of pieces on the slide here. First, I put the water, maybe PBS and so on, buffer. So pick up small amount of pieces and mixing and spread the pieces and put the cover glass here. So trophozoite can survive maybe for 2 or 3 hours. That's why we had better check the trophozoite immediately after collecting the pieces. But instead of trophozoite, most of the parasites can survive for a long time, but it depends on the species of the parasite. For example, you must note the viability of the parasite. Oocyst or cyst can survive for a long time, at least several weeks to 1 or 2 months. So we can keep these feces or parasite at the refrigerator for a long time without any adding reagent, ethanol, methanol, and sodium gchromate, and so on. We don't have to. And eggs of nematode or cetode and so on can survive for a while, although the viability can differ among the species, but almost it seems to survive for 1 to 2 weeks or months, several months at least. So in such target parasite, we can keep fecal samples at 4 degrees, of course, in refrigerator, but not being frozen. We don't have to keep in under 20 degrees. So we can keep these feces at 4 degrees in most cases. Why? Actually, freezing treatment can kill the parasite. This means changing of the specific gravity and reduce the sensitivity of flotation method. Of course, if we freeze the fecal samples, we can kill many parasites, but the sensitivity of the examination will be degrees. But if you can take DNA from the feces directly, we can freeze feces. Of course, it's okay. Additionally, we do not add any reagent, usually for a short period storage. For example, formalin, formaldehyde. In some countries, researchers added formaldehyde, but formalin can inhibit PCR reaction as additional analysis. That's why I recommend we don't have to add any reagent for several weeks. This is the protocol, summarize of the protocol for examination. Of course, we will collect feces in most cases. So as a method, first, direct smear, we can diagnose most of the parasites, but we do not recommend in low OPG or EPG samples because we just pick up very small amount of feces. That's why this direct smear can be low sensitivity, but very easy. So segmentation method can be diagnosis for Trombetode or Baxtinella and so on. Then, in flotation method, we can see many species of the parasite. For example, nematodes, cetodes, oocysts, and cysts, and so on. That's why we can easily or generally apply flotation method for diagnosis of these parasites. But you must choose the objective, including information about the biology of the parasite. Then we collect the feces here, so we can separate instead of except for directive smear, flotation method or sedimentation method. Of course, if we can do sedimentation method, we will diagnosis about Trombetode. Trombetode, egg of Trombetode can be settled down at the bottom of the tube here. But flotation method, as I explained, we can check many species of the parasite. In flotation method, usually we use two types of the solution. One is saturated saline solution. The other is sucrose. In some cases, sucrose is a little expensive, but we can use the sugar instead of sucrose. So you can easily buy gula at the Alphamart or Indomart easily. That is why instead of saturated saline solution or sucrose, we can use sugar or gula. So we can check many species of parasites. For example, protozoa, nematode, cetoda, and so on. So, can you identify the parasite morphologically? Can you identify the parasite based on the morphology of the egg or cyst or oocyst? Actually, it depends on the purpose. So, why? I will explain the answer. The parasites are sometimes very small and difficult to be detected. So the parasites are sometimes difficult to be differentiated from other debris. In feces, we can easily so many debris which are not parasites. That's why we must differentiate. This is a parasite or just debris and so on. We must differentiate. And the parasites are almost impossible to morphologically identify species. For example, this is an egg. If we see from here, maybe the shape will be round. But if egg like this, we cannot see the round. Maybe subspherical like this. That's why the side of the seeing can be changed the morphology, round and subspherical and so on. That's why it's sometimes difficult to identify based on the morphology. I often found that some authors identify the protozoa under microscope by mistake. For example, in some papers, this is oocyst or and so on. But in my opinion, these are not oocyst. So that is why, for example, student said, this is a cryptosporidium. But I answered, what's evidence? This is round and small. No, these are enough information for identification. You must just say, this may be cryptosporidium, like this. So sometimes difficult to differentiate parasite and debris, like here. For example, these are not parasites. This is residue from plants here. Very rough inside here. This is not parasite. This is not parasite, of course. They are yeast because we cannot see any contents inside. And this is a spore. because no contents here. This is just bubble air because very round and thick form, very dark shape. And this is the front. Why? Because in most cases, the egg of parasite or oocyst and cyst is the same size, almost the same size and the same shape. But here, small one, large one, and bigger one, different size. In such a case, they are not parasites. And this is the front here. So this is also plant cells. In many cases, we can see the contents of the cyst or oocyst and egg of the parasite. But we cannot see any contents here. But in the egg of Ascaris, sometimes very dark, but we must take care of the Ascaris egg. This is also plant cells here. In plant cells, the walls are very thick and very dark. But in parasite, dark wall means zoetes cannot release. That's why very thick wall means not parasite in most cases. And this is the pollen from the pollen plant. Maybe host ate these plants in the field. That's why in the feces we can easily these plant cells. But we can differentiate based on the shape or color. This is very dark, and this is too thick here, and informal shape like this. This is not parasite. And this is the thick wall and no contents here. And this is also the too thick wall like this. That's why the textbook only shows a nice picture of the parasites, of course. But in the field or in the microscopic observation, you must differentiate by yourself. That's why, of course, you must remember the shape of the characteristic of the parasite. And then we must examine the parasite under microscope. But, okay. For training, it's good to ask the specialist, of course. So, how to identify the parasites? We must know well how the parasites are morphologically seen on the slide. What's color, or cyst, or cyst wall, or contents. We must understand first. Then, how the parasites are confirmed. For example, we can do sometimes specific staining or immunostaining. They are sometimes very helpful if you can do. However, only morphological data are sometimes risky to be identified exactly and can lead misunderstanding of the infection in the host. In case of the research, we had better diagnosis exactly the parasite, but not in every cases. For example, in the field, we don't have to identify the species because we can use the same therapeutic drugs. For example, if we find Eimeria bovis, Eimeria tsuerni, and Eimeria alabamensis, but we can use only the same drugs. For example, Bicox or Tolteroduril, and so on. In such a case, we don't have to identify the species. But in case of the research, or in the case of surveillance, we must diagnose or identify exact species. So case by case, it depends on the objective of the examinations. If you want to identify exactly how should we do? You should try to analyze by molecular method. So the second topics here from here, I will talk about basic literature first of molecular analysis of the intestinal parasite. How to analyze a parasite by molecular tool. Of course, you cannot do immediately, but if you understand logically and generally, so you can do in the future. So for your future analysis, it's better to understand logically. So this is the summary of the molecular analysis. I just pick up the name of the molecular analysis. As you know, PCR, nested PCR, RT-PCR, real-time PCR, or RFLP, or PCR-RFP, PFGE, LAFD, or and so on, microarray or lipotyping. But usually, we can easily try the method of the first one, PCR, nested PCR, and so on. PCR methods are frequently used worldwide for identification of the parasite. Basic PCR protocol in this slide. So first step, we must prepare for DNA template from the parasite. And we had better establish PCR condition, including primer design and formula of the PCR reaction and so on. And number three, third step, PCR and gel electrophoresis to determine the PCR product. And if you need, we can do sequencing and data analysis. These are a step of PCR, a basic PCR protocol. But this PCR protocol is not always easy because in parasite can be special.
Dasarnya masakan.
So,
sehingga kalau seperti itu pasti akan risiko pendarahan akan.
Every step are very important for the parasites. I will detail or careful point. First step, we must prepare of the DNA template from the parasite. It's sometimes difficult to prepare for the DNA of the parasite. For example, we can separate helminths and protozoa. In case of helminths, helminths possess multiple cells, means so many DNAs. So, for example, in helminths, we can see nematodes, trematodes, cetodes, and protozoa. In case of nematode, trematode, cetode, we can pick up easily adult worm and sometimes larvae. So these huge parasites possess many DNAs and easy to pick up by pincet and washing. And they can be identified by morphological data. But in case of eggs or protozoa or cyst and cyst, of course, adult and larvae, very easy. But egg of these helminths or protozoa like this, mainly single cells of one nucleus, means very huge amount of DNA will be contained. In such a case, these protozoa and egg of helminths are difficult to purify them from feces. Maybe during purification of the parasite, we will lose the DNA of the parasite, like this. And they are difficult to extract DNA from egg or oocyst. Why? DNA contains in egg or oocyst or cyst. But the wall of egg or the wall of oocyst are very strong and robust. That's why we must break the oocyst or egg, and DNA will be released. That is why sometimes difficult to extract the DNA from egg or oocyst. But especially protozoa possess only one nucleus here. That is why small amount of DNA will be included. And many DNA of other organisms in feces, from bacteria or host cells and so on. There are so many other DNAs. And PCR inhibitors will be contained in feces in most cases. That is why in my imagine, this is, for example, feces here. And this is like universe because bacteria, there are so many bacteria, and genome size will be 210 megabase pair. And in case of host cells, one cell contains 2.9 gigabase pair, and in pig 2.7 gigabase pair. In chicken, 1.8 gigabase pair will be contained. But parasite will be 10 to 100 megabase pair. In such a case, of course, the DNA of the parasite will be included, but so many other DNA will be contained. We must be careful in this point. So, first, in case of large parasite, of course, for example, this is Ascaris sum. We can pick up only one worm like this. Easily, we can pick up and washing and cutting or breaking easily. So we can easily extract the DNA by phenol chloroform and ethanol sedimentation method, or this kit. Kit will be a little expensive, but there are so many merits. These kit will be less toxic because phenol chloroform will be very high toxic, and this kit can be less toxic and easy to extract the DNA, and it takes short time like this. But as a demerit, these kits are expensive and sometimes loss of DNA. But we recommend the use of this kit for extraction DNA if you can. But these large parasite will be easier compared to small parasite. Then, in case of small parasite, egg or oocyst here, of course, if we extract the DNA from the feces directly, we can keep the feces at 4 degrees or minus 20 degrees. They can be okay if you use feces directly, like this. So we can use here again DNA struminicate or DNA Zol direct or DNA Zol or others. So if we pick up the feces for the extraction of DNA, this is a merit. This method is very easy and preserve for a longer period at minus 20 degrees. And this method is suitable for trophozoite because we cannot purify the trophozoite using feces. And demerit, we just use 200 milligram in most cases of feces. This volume is very small. That is why we can extract the DNA at the small volume. And DNA are not released from the robust egg or oocyst in this protocol. in such instead of this protocol, how we do the extraction of DNA. This protocol is very useful for us. In most cases, we use this protocol. First, we use huge volume of feces. For example, match feces here, and we can keep 4 degrees. And we dilute the feces with water like this. And we centrifuge this tube at 2,500 or 3,000 RPM, 4 to 10 minutes. And but we can do filtrated these feces or ethanol treatment to remove the lipid and discard the supernatant. We added the flotation of solution, for example, sugar solution. This sediment gravity is 1.2. And then I centrifuge again. So most of the parasites can be floated here like this. And then we aspirate the supernatant upper layer like this. So around here, many parasites can be concentrated. So we pick up only the parasite like this. So after purification of the parasite by sugar flotation method, we collect like this. But not only a oocyst or cyst and egg of the nematode. So it contains many bacteria or host cells can be contained here. But we can concentrate the parasite here. So only oocysts can be treated with hypochlorite acid. This is a new protocol that we found previously, because hypochlorous acid can be killed other DNA or microorganism. So, but oocysts can be survived even in hypochlorous acid. Oocysts can be survived in such a strong sterilization solution like hypochlorous acid. So the treatment of hypochlorous acid can reduce other DNAs. So PCR, the sensitivity of the PCR will be increased. Before the extraction of the DNA, usually we added the hypochlorous acid. And then we must disrupt the parasite from the oocyst or cyst and egg of the hermes. So how to disrupt the oocyst or egg? Usually we use the freeze thawing or glass beads. We added glass beads and vortexing, and beads disrupt the oocyst like this. So DNA will be released from the parasite. And then we can do the we can use the this extraction kit. That is why we have to better use purification first, and second hypochlorite acid treatment to reduce other DNAs. And third, we had better break the parasite to release the DNA. That's why we just use a key again or a DNA extraction kit. We must do 3 steps at most. So this is PCR conditions. Maybe you can see easily in many published papers. So in this formula, the most important factor can be primer, as you know. So we must select or design carefully the primers. depending on the purpose. So this is the PCR conditions. You can easily understand maybe here. So this is the very, very basic protocol for PCR reactions. As you know too much, this is a template. First step is denaturation. And second step is annealing. So primer can be hybridized to the template. And third step, the extraction, extension like this. And we can do repeat usually 35 to 40 cycles here. So in this protocol, of course, design of primer are very important. And we must design them depending on the purpose. So I will explain how to design the primers to diagnose the parasites, especially. So this is sequences or known sequences like this. And usually we designed forward primer and reverse primer by PCR. So the size are effective as long as possible, generally. But longer size tend not to be amplified easily, and sometimes difficult to be sequences in case of longer PCR product. Because if we sequence the PCR product, of course, we can obtain the sequence data by forward primer and reverse primer. So we had better check both sides, the sequencing. That's why we can do overlapping, because we can check easily for two directions. That's why we had better overlap for sequencing. Based on the evidence, the size of the PCR product can be effective in 500 to 800, 100 base pair. It might be a good length for PCR products. So how to design the PCR primers? For example, this is a non-sequencing here, for example. And we will design forward primer and reverse primer. Then genotype, this is the sequence of genotype A. This is the sequence of genotype B, and genotype C, and genotype D. So these regions are conserved regions, almost the same in these regions. But this region can be different among the genotypes. This is also, if you design based on different sequence here, like this here. So if you choose low conserved regions, different regions, you might construct species, genotype A, specific PCR, of course, because these primer can be hybridized, genotype B, C, D. This primer can be hybridized only to genotype A. That's why you might construct the species or genotype A specific PCR. But it could be difficult to show complete high specificity because we cannot check easily all of the genotypes for PCR analysis. That's why we cannot obtain all of the species or how to identify two. Because if you amplify these species, but this region can be the same among the genotypes. That's why we cannot differentiate. It's very difficult to differentiate based on these regions. So how should we do? You have better select conserved regions to make primers. For example, This is genotype A, B, C, D. These regions are different among the genotypes. So we select the same regions for making the designing primers like this. In such a case, if you choose high conserved region for primers between low homology like this, so you can identify the species genotype by sequencing because these regions can be changed different. That's why this sequence region, if you can obtain these regions, we can blast search. You can do blast search, and you can determine exactly the genotype A like this. So you can find new other species in various hosts and so on. So, but we must take care of mixed infection. For example, in case of cattle, pig, and chickens, if you collect the feces from the host, but in most cases, there are so many parasites in the same host. For example, usually these parasites can be mixed in feces. So if you purify the parasite and extract the DNA, and if you can do PCR, but in the case of the infection, mixed infection, you can see mixed sequencing like this. In such a case, you cannot determine the sequencing. So in such a case, very difficult to identify the parasite. In such a case, in our laboratory, using the manipulation, so I pick up only one oocyst based on the microscope. So I pick up one oocyst and I pour one oocyst into PCR tube, and we just do freeze thawing and boil at 99 degrees for 5 minutes. And we can use the samples as a template. So we can successfully obtain the sequence. For example, like this. This parasite, the sequence of this parasite will be here, and so on. So in case of mixed infection, we had better pick up only one oocyst to identify. So it's so nice if you find the published primer pairs. It is sometimes so difficult to design by yourself. In such a case, you had better check published primer first. So if you find published primer already, so you can easily apply these published primer. You don't have to design by yourself. So, but you have better check the protocol carefully. So this is a methodology. Protocol to check the published primer first. It's better for you. First, please check the primer set among published papers first. If you are lucky, you can reduce much effort to design primers. And second step, please check the primer conditions on these papers. For example, if PCR product size is very huge, it is difficult to sequence the PCR product. In such a case, please reject the published papers. So if you can select the other published papers. And please check target stages of the parasite. Only for adult worm or only for larvae. or only for the egg and so on. You must check the target stage of the parasite. And you had better check how to purify the parasite in published papers. So, and finally, you had better check species-specific primer or not, and so on. Next slide. Sorry, please wait. Okay. Okay. Number three step. If you cannot find any papers, or you cannot find any published primers, so in such a case, you must check deposited sequence on gene bank. So you can finally design the primer by yourself based on deposited sequence. If you cannot find any deposited sequence in gene bank, it's very, very difficult to design the primers. That's why first step, you had better check published paper or published primers. And the second step is checking the deposit sequence in gene bank. If you find the sequence successfully, and fortunately, you can design primer by yourself based on gene bank sequencing. So, but you must check the regions of the sequence. If the region of Hugh data, you cannot compare with other species or genotypes. And if you usually you can use the region of 18 small ribosomal RNA gene regions, or cytochrome C regions, ITS regions, and so on. You can easily find these sequences. But if you can find only very rare regions, it's very dangerous to analyze furthermore, because we cannot compare obtained sequence to other many species. We cannot compare. That is why we must check the region of the sequence in addition to the sequencing. So finally, as the third topics, I will introduce my recent and very new findings. This is not yet published. Actually, my student is writing the paper, but not yet. Maybe I will submit the paper immediately after a student complete the writing the paper. So the title is The Detection of Intestinal Parasite in Nutria, Mycastor coipus, and Molecular Identification of Strongyloides myopotami from the egg. Do you know the Nutria? Can you find the Nutria in your country? I guess Nutria can be spread in the world, especially in Japan. The Nutria will be imported from other countries, and the number of Nutria will be increased. So, like this here, very cute, but they ate many crops. That is why Japanese government pour much effort to reduce the number of Nutria.
But in the South America, I think so many.
Yes. How about in Indonesia?
Nothing.
Nothing.
Yes.
Oh, okay.
That's not famous, you know.
Yeah. Very lucky.
Yeah.
Okay, I will introduce what is a Nutria. Nutria, the taxon of Nutria, Mammalia and Rodentia, Mycastoridae, like this. They inhabit close to the river and like a beaver, but not so huge. The origin of the nutria is southern area of South America. So South area, at the South area, original or origin of the nutria. So about the distribution, they can be currently colonized around the world, and after human-made translocation, human moved the nutria from South America to Japan and so on. So habitat, the nutria inhabit near lakes and ponds and streams and swamps. So they are called as semi-aquatic animals. Of course, in the river and in the ground, they can move to both. And in fact, they can show damage on agriculture and aquatic plants. They can eat so many plants, corn and other crops, and destruction of bank and rice field and walk and water reservoir. They can easily break this field. So in the past, they could use as fur or meat, but now we don't use nutria. So in Japan, nutria, the number of nutria is increasing, but not North area, around here. Of course, Osaka is included here. Of course, Yamaguchi also.
Yeah.
So unfortunately, the nutria can bring
so many
so many pathogens. For example, parasite, bacteria, virus, and rickettsia, and so on, others. My student searched many pathogens by many published papers. So he picked up one by one. So we were very surprised that nutria could bring so many pathogens. But and red color means zoonotic pathogens. So the increasing number of nutria means they can bring so many pathogens to us, human. So red color is zoonotic. So we must take care of, pay attention to the nutria. So I will focus on this parasite, Strongyloides species. In nutria, in the world, some researchers reported the infection of Strongyloides myopotami in nutria. This Strongyloides myopotami is the nematode Raphiditaria strongyloidea type. And the parasite, the host of the parasite is nutria. But many information remains unknown. For example, infection route are unknown. Symptom is unknown. We don't know, or we don't have any information. And for diagnostic method, researcher cultivate the egg for larvae, and after catch the nutria and kill the nutria and open the intestine, and researcher could pick up adult one for catching the nutria. And zoonotic potential. Actually, in human, severe cutaneous rash by skin penetration or larvae could be occurred in human. That's why this parasite can be zoonotic, like this. So in our research, objective on this research is fecal examination of intestinal parasite in nutria, and to assess a zoonotic risk to human and other animals. And second, development of sensitive molecular method using eggs in feces to identify Strongyloides myopotami. These are two objectives in this research. So this is the material and method. This slide shows the samples. Location of samples, Osaka Prefecture. I pick up 5 fecal samples of nutria. And Okayama Prefecture, we pick up 8 fecal samples. Osaka is here, as I explained previously. And Okayama is here. Okayama will be future metropolitan cities. So we targeted 2 prefectures in this research. So total, I collected 13 fecal samples in this research. Terms is between 2020 and 2021. And the information of nutria are unknown about sex and ages, no information. And fecal samples were kept below 4 degrees, not freezing. So the number of nutria in Osaka prefectures and the Okayama are very increasing, like this. And most samples were load killed or captured nutria to reduce the number of nutria. So this is a protocol for fecal examination. I suspend 1 gram feces with water and remove debris by filtration with Gaussian and centrifuge again, and dilute with sucrose solution, specific gravity 1.2, and centrifuge again. And I collected upper layer of the parasite by Nicron loop, like this. This I made the Nicron loop by myself. And we examine the slide glass under a microscope, like this. So in case of positive samples, we purified the egg of Strongyloides myopotami by flotation method. And I did freeze thawing, and breaking by bees. And DNA was extracted by Kiagen kit. So this is the result and the prevalence of egg. And this is the number of samples. And we collected total 13 samples of nutria. So we could find nematoda egg like this. This is an EPG, egg per gram, or egg number of 1 gram feces. So we could find egg in 12 samples. Number 6 are negative, like this. But the other samples were positive. And in addition to nematoda egg, we could find Eimeria species, like this. But in this research, I focused on nematoda analysis. So, as I explained, how to do PCR analysis. First step, we must check published primer. So I checked the papers analyzing the nutria nematoda. So fortunately, I found published primers, F1, F2, F3, F4, F5, 5 species of forward primer. And R1 and R2, two type of reverse primers. So this is the position of the primer. Actually, this is the example sequence of Strongyloides sterocolaris. So in this sequence, the most important region will be HVR1, HVR2, HVR3, HVR4. Actually, HVR means hypervariable regions. Means we can differentiate the species based on HVR1, 2, 3, 4. Because among the species, these regions will be almost the same. That's why we can differentiate based on the specific regions. That's why, for example, F1 and R2 contain 4 HVR regions. Can you understand? So F2 and R1 contains HVR2, 3, 4, these primers. That's why, based on the sequence of 4 variable regions, we can differentiate the species like this. So first step, at the first step, we could try these published primer first. So this is a result for published primer and sequencing. We use number 7, 8, 9 of Nutria. So this is a primer set. F1, R1, F2, R1, F3, R1, F4, R1, F5, R1. This is a combination of this one. F1, R1, F2, R1, and so on. So this is expected amplicon size. This F1, R1 is very long, 1,500 base pair. For example, this F5 to R1 is 300 to 20 base pair. This is the expected amplicon. Yes means we can see expected amplicon. For example, yes means we could see 1,500 base pair amplicons. So, for example, in three samples, we could find only one samples in 100 base pair. Like this. Yes, and so on. But we did sequencing analysis. For example, in case of F1, R1, so we could see expected amplicons. But by sequencing analysis, obtained sequence could be derived from Pseudomonas. Or F2, R1 means bacteria, and so on. That means these published primer could amplify the DNA from the bacteria. So, as I explained, in feces, so many bacteria DNA. That is why these specific these primer can be amplified the DNA of bacteria. And additionally, F1, R2, F2, R2, and so on. Of course, we can see yes. But in a few cases, we can determine Strongyloides myopotami. This primer set seemed to be good, but like this, non-specific band could be seen in case of primers. So published primer can be good for adult worm or larvae of the Strongyloides myopotami. So in case of eggs from the feces, these published primer cannot be suitable. That's why we determined to design by ourselves. So this is the published primer information. So this is the information of our designed primer. For example, I named ST-Mayo F1 R1. So HVR1 and 2 and 3 could be contained. And F2 and R2 contain HVR2 and 3. FC-Mayo 3 and R3 contain HVR2 and HVR3, like this. These three primer pairs were designed by myself. So the length of the PCR product were 500 to 700 base pair. So this is the result. I used the sample number 7, 8, 9, only 3 samples. And ST-Bio F1 R1, F2 R2, F3 R3. This is the expected amplicons. So after gel electrophoresis, we could do the specific band in all primers like this. And the sequencing could be identified like this. Strongyloides, myopotami, myopotami, myopotami. And in case of non-specific band, we could not find any non-specific band, except for 1 cases. That means we designed this primer pair is good, can be good for the egg of Strongyloides myopotami in feces. So the next step, we evaluate the sensitivity of 1 egg or 10 egg, very few number of eggs. So F1 R1, F2 R2, F3 R3. I pick up only 1 egg and 10 eggs. So 3 samples of 10 eggs, we could successfully obtain the expected amplicons in all, like this. This is 10 eggs. This is 1 egg. We tried 5 samples of 1 egg, but in 4 samples, we could successfully obtain the amplicons. So at least we could amplify only 1 egg of Strongyloides myopotami. So the development PCR assay were able to detect even 1 egg of Strongyloides myopotami eggs, like this. So actually, in addition to determination of Strongyloides myopotami, we try to determine the haplotype of Neutria. If we can determine the haplotype of Neutria, we can analyze the distribution of Neutria. For example, in Okayama Prefecture, haplotype A Neutria can be spread. In Osaka, haplotype B of Neutria can be spread, and so on. Usually, other researcher analyze the haplotype using the samples, ear or blood, and so on. But I tried to determine the haplotype using feces. So actually, we can determine haplotype of Neutria using feces. So if we collect the feces of Neutria, we can check the infection of Strongyloides myopotami and the haplotype of Neutria, haplotype A and B, and so on. Based on the protocol, we can evaluate haplotype B could be infected highly with Strongyloides myopotami, and so on. So far, DNA was extracted from liver or ear with sacrifice to determine haplotype of Neutria. But in our research, in the previous study, we could determine haplotype using small amount of feces. So, conclusion. The most important first step is observation under microscopy by exact method. For example, direct smell or flotation method or sensitization method. This is the first important step. So we had better know, but it's sometimes difficult to identify the species morphologically. For example, the oocyst of Eimeria is sometimes difficult, like this or like this, but the same oocyst. So, but it's okay, depending on purpose. So PCRs are not always so easy if we do not understand the detailed method, how to purify, how to disrupt the oocyst and egg, how to extract the, collect the DNA and so on. PCR is very easy, but in case of the parasite, sometimes we must take care the important steps. If you want to learn, if you want to learn and to be the specialist, we are so welcome that you will come to Japan or be a PhD student. So, of course, you are the candidate of good veterinarians. Maybe you will or you must contribute to the high productivity in your country, or to save or control the infection in pet animals. So to complete the contribution, we should exactly diagnose the parasite. So this is the final slide. I'm really enjoying my talking, and I'm really appreciating having a lecture to your students. Thank you very much. I will send my best acknowledge. Terima kasih banyak. Thank you very much.
Okay, thank you, Sensei. Thank you, Prof. Makoto, and nice presentation. So this is the good opportunity to explore more about maybe some question. If ladies and gentlemen, you can share your question by chatting room. Okay, please. And then so there is some good answer from the incredible lecturer from Osaka Metropolitan University. I think this is very good time to learn more about some how to detect, how to collect, how to identify by the molecular identification, something like that. Particularly in the nutria, is the good information for our people. Even the Indonesia not so nutria, And so I think this is for everyone. If there is some question for to Professor Makoto Matsubayashi. Okay, please. If there is some question, particularly for master student or doctoral student and also PPDA student. So if there is some question, because this is some the new one about the information about the potential reservoir of nutria, look like red, because the ecosystem and also environment in the rice field, and then some of the poro, something like that, particularly in the what's called it, maybe some the vegetable. So sometimes we find some strong like this, stercorale, something like that. So that in the environment of nutria, I think so link and match to be some reservoir, some strong like this myotomy. Yeah. Okay, please. Okay, now is there is question from BB body fat from Dia. You know Dia? Any question? May you explain the presentation of sodium hypochlorite usually used and how to use the eliminate before extraction? Sensei, this is the question from Dia. Presentation of sodium hypochlorite.
Yes, hypochlorite
Yes.
is good to
yeah.
to kill the other microorganisms in feces.
I see.
So oocyst, so after treatment of sodium hypochlorite, so
Yes.
only the oocyst
Yes.
can be survive.
Yeah.
And if of course after purification of the oocyst by sugar flotation, so the solution, the color of solution is brown. But after treatment of hypochlorite, the solution, the color of solution can be changed as white.
Yes, white.
That is why the oocyst, the color of oocyst could be white. So hypochlorite remove or kill the other DNA of microorganisms.
I see.
So in case of the PCR, non-specific reaction can be reduced.
Yeah.
But if you do the treatment of sodium hypochlorite, you have to better wash at least 5 times.
Yeah, I see.
Repeatedly wash and centrifugation and discard and water washing.
Yeah, that is very important to eliminate some hypochlorite, sodium hypochlorite. And then how many concentrations, Sensei, if we use oocyst purification?
About
about some the sodium hypochlorite.
Okay.
Sodium hypochlorite.
In my case, I use 10% of sodium hypochlorite.
I see, 10%.
But you can use 1%.
I see.
Sodium hypochlorite is very strong solution. That is why, in my opinion, 1% of sodium hypochlorite can be enough.
I see. Yeah. 1%, you know. It can be enough.
Oocyst can be survived, but cyst or egg of nematoda can be killed together.
I see. Yeah.
That's
even the wall is thick, you know. Yeah.
Yes.
Yeah.
We must take care.
Prof Yunus, I have question.
Yes. Okay. Please, your raise hand by reaction. Okay.
Prof Makoto.
Yeah. Dr. Apel will ask you about some question. Okay. Please. Maybe Dr. Nunu or Prof Nunu.
Yeah.
Okay. First, Prof Nunu. Okay. Please. Say.
Thank you very much, Prof Yunus, as a moderator. Thank you, Prof Makoto. You have nice presentation about the molecular technology to diagnosis of parasites, especially parasites in gastrointestinal of livestock. I want to know how many sample minimal should be sequenced to use phylogenetic analysis. I want to know in the analysis how many percent homology with the other species in the gene bank. I have research, for example, entamuba, identification of entamuba poleki in East Java with the sequence, and we analysis with phylogenetic tree and so that the poleki in East Java close with poleki in human subtypes 3.
Yes, we can select some samples depending on the situation. For example, if you survey 10 farms, So we can extract one sequence each palms and so on. Or you survey 3 prefectures. So you can sequence 1 or 2 samples in each prefectures and so on. It depends on the situation of the survey.
Okay, 3 sample. Okay, thank you very much.
Okay, the next question from Dr. April. Please, Dr. April, the time is yours.
Thank you very much, Prof. Yunus.
Yes.
Good morning, Prof. Matsubayashi. How are you?
Morning. I'm fine. Thank you very much.
Thank you very much for.
Best friend. Best friend.
Yes, thank you very much for your constructive and comprehensive presentation. I enjoy your talk. It's very nice. Thank you for show the result that you already conducted there. It's very interesting. I have some question regarding your talk. The first one is about using one OOKIS for DNA extraction. To be honest, it is very challenging. We pick up only one OOKIS and continue and followed by the DNA extraction.
DNA extraction.
Yeah. Perhaps you can share for us how do you do technically in the laboratory when you pick up the OOKIS. And as you know, for example, in chicken, there are 7 Emeria OOKIS, right? And then it is difficult for us to identify them based on morphology. So, in the case to pick up the OOKIS, sometimes we are not confident. This is the Emeria tenella, this is the cicatric, or this Estropolina, because they look similar sometimes. How we select them? Okay, this Maxima, I will pick up one, and there's the Arcifolina, I pick one, something like that. How we select them? How we distinguish them from other species? And the third one, it is about sodium hypochlorite that you use. You mentioned that the sodium hypochlorite can eliminate bacteria if we want to extract OOKIS. How about egg of helminth? Is the sodium hypochlorite also eliminate the egg, or just only bacteria? And you just said that if we use the sodium hypochlorite, we have to wash 5 times, right?
Yes.
It means there is a possibility for us to reduce the DNA extraction product. How we deal with that? How we anticipate for losing the DNA extraction using sodium hypochlorite method? Thank you.
Thank you very much.
Okay.
Thank you very much for your nice questions. First question is about how to pick up one oocyst. So if you want to pick up one oocyst, for example, please prepare for the slide, and you pick up 5 microliter from the purified solution of oocyst and put on the slide. Put on the slide, and check the microscope.
Yeah.
Under the microscope. And one oocyst, it's okay. Please pick up and chew. And if 2 or 3 oocysts can be seen, discard. Like this.
I see. Yeah.
1 candidate method.
Okay.
And about the Amelia maxima or tenera and so on, if you want to identify the species of one oocyst. Actually, in chicken Amelia, we can use species-specific primers.
Okay.
So in
I
such a
know.
case, we can use mixed DNA. That's why in case of chicken Amelia, we don't have to pick up one oocyst. Fortunately. And in case of egg of nematoda, actually I and my student pour sodium hypochlorite to the egg of nematoda and washing 5 times. We check the we check the egg of nematoda under microscope. So all of the egg were disappeared.
Okay.
So only the oocyst can be available.
Okay. Thank you very much, Professor Matsubayashi.
Thank you very much.
Yeah. Okay. The other ones. Sensei, I continue Dr. Apel question. I think to make sure when we pick up one oocyst, to make sure this is already picked up or just to disappear after we pick up from the slide, and then you move to the tube, I think. Yeah, this is very difficult to make sure this is already picked up or not, because one oocyst is very small, even the chicken. How to make sure about it?
In such a case, for example, 5 microliter. So
yes, I know. Yeah,
we put so many spots.
Yeah, I know.
And checking, and this is only one oocyst. And pick up.
Yeah.
And please pour one oocyst to the PCR tube.
Yeah.
But of course, after pour the one oocyst to PCR tube,
yes.
Unfortunately, we cannot confirm.
We cannot see anymore.
We have better pick up so many tubes.
I see.
And PCR, and maybe 10 tubes, and maybe 3 positive PCR.
Yes.
Okay.
Good strategy, Sanjay. Good strategy. Yes. Yeah, I know. Okay. Any other question from the audience?
Yunus.
Maybe,
yes.
Prof. Yunus.
Okay. Professor Lucia. Okay, please. The time is yours to give us question from Professor Makoto.
Thank you, Prof. Makoto. Thank you for the great presentation. Allow me to ask one question. In your experience for detection strongyloides, is it better to use eggs or larvae?
Larvae.
In your presentation now, you use eggs strongyloides. It's
which one is better, correct? Between larvae or the eggs?
But it's better. Is it better to use eggs or larvae?
Larvae. Larvae.
Yes. Then say, Professor Lucia said, which one is better? We take some larvae or the eggs of strongyloides? Which one is better? To take larvae or the eggs?
Sorry, what is the larvae? Larvae?
The young strongyloides. The youngest strongyloides. Just emerge from the eggs.
From the egg.
Or the eggs.
Just
from the egg, yes.
Yeah, just hatch from the egg.
Yes.
Egg.
So in published primers can be available for adult and larvae.
I see.
So in case of the egg, published primer could not be amplified.
Oh.
So because larvae or adult worm could be picked up and less bacteria. But from the egg, egg could be contain so many bacteria.
Yeah,
That is
yeah,
why non-specific one can be visible.
yeah.
Larva.
Yeah. Okay. Okay. Any other question?
Thank you.
Yeah, welcome. Any other question?
Can I add some one question?
Okay. Please, Dr. April.
Yes, you said that we cannot we cannot do sequencing for egg of Strongyloides because there are so many bacteria on the DNA extraction. How if we successfully design the specific primer for the egg? Is it possibility? Is it possible for us to sequence the DNA of Strongyloides from the egg if we successfully design the specific primer for the species? How do you think?
Yes, if we want to design specific primers, but unfortunately, we can confirm specificity because if we design specific primer, but we must check with other species of DNA, but we cannot prepare for the other species DNA. It's very tough. For example, I design the primer, specific primer for Strongyloides myopotami, but we must check other Strongyloides species, Sterochlorias, and so on, we must check the specificity using other DNA, the DNA of other species. It's very tough to collect the DNA of other species. That's why we logically we can design the specific primer based on the sequence, but we cannot confirm using the DNA of other species.
Thank you.
Okay. Any question from the others? Okay. Okay, Rakil, are you? You want to ask something? Rakil, are you? Because in your profile, like your right hand. Rakil, are you? Are you present here? Okay. Any question from the participant? Sumo?
Maybe.
Okay.
Maybe one student.
Oh yes. Okay, from Sally. Okay, please.
Good morning, Professor Makoto. May I asking you, you mentioned that formalin can inhibit PCR. How about another solution like alcohol or ethyl alcohol? That's more better or not? Thank you.
Oh, it's so good question. As I told, formaldehyde can inhibit the PCR reaction, but ethanol is better. Ethyl alcohol can be good for the preservation of the parasite.
I see. Yeah. Okay.
Thank you, Professor.
My pleasure.
Okay. We still have time, maybe around 2 minutes or 3 minutes if there is any question from the other, from the participant or audience. Okay. Okay, if there is no question more. Okay, thank you, Professor Makoto. So your presentation is very benefit for us to learn more about the molecular identification and also the technique of the good the process of how to take the new information about the nutria potential reservoir for our zoonotic disease, I think, because so many some the isolate pathogen from the nutria, you know. Okay, the session of discussion, and I think that's all. And then everything about the presentation and also the lecture from Professor Makoto Matsubayashi. Thank you very much once again for your nice lecture today. Okay, is as a moderator, if there is some mistake from me, just I apologize for that. So now is I return back to MC. Please, Dr. Rania. Thank you very much.
Yeah, thank you very much, Professor Yunus. So once again, thank you for Matsubayashi Sensei for the insightful lecture and for sharing new knowledge together with us. And also thank you for Professor Yunus for acting as a moderator. And once again, we will have a virtual photo session before moving on to the final session. So for all participants, please turn on your camera. Okay, so for the first slide, we will start the countdown for the first slide in 3, 2, 1. Oh. All right, so for the second one, we will start the countdown in 3, 2, 1. And for the third slide, we will start the countdown. 3, 2, 1. And for the fourth slide, 3, 2, 1. And for the fifth slide, 3, 2, 1. And for the last slide, 3, 2, 1. Okay, once again, thank you for all participants for participating in this guest lecture. And the final session will be assisted by Dr. Endang Supriyati, DVM, Master of Science. For Dr. Endang, the time is yours.
Masih mute, Dr. Endang masih mute. Dr. Endang masih mute.
Assalamualaikum warahmatullahi wabarakatuh. Good afternoon and warm greeting to everyone. First of all, special thanks to Prof. Makoto Matsubayashi as our remarkable speaker today. Therefore, on behalf of the Division of Veterinary Parasitology, Faculty of Veterinary of Medicine, I would like to hand over the certificate from Prof. Makoto Matsubayashi.
Thank you very much.
This is Prof.
Okay.
Yeah. Thank you very much, Prof. Makoto Matsubayashi. Already share your experience and valuable knowledge to all of us in this quiz lecture event. Hopefully, we can meet directly and conduct more collaboration research and scientific publication. And now, the guest lecture session is finished. Personally, I would like to say thank you to our speaker and all of participants, including all student, lecturer, and sitter, researcher. Thank you for joining us today. I do apologize if there are any mistake or technical problem during the guest lecture. Therefore, I would like to close this guest lecture even at the time. Once again, thank you very much. Hopefully, all of you will be gain many new knowledge and have a great day, everyone. Okay. Good afternoon. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Big applause for Makoto Sensei. Big applause for Makoto Sensei.
Ya selesai. Terima
And
kasih.
you got
Thank
say
you
hello
very
from
much,
Tito.
Prof. Makoto.
No, Tito.
Thank you,
Tito.
Prof. Makoto.
Thank you. Halo
See
Makoto.
you next year.
Matur suwun,
Thank you.
Prof.
Silvia.
Sami-sami.
Teman semuanya. Terima kasih.
Oke,
Kak
terima kasih.
Diana.
Thank you very much.
Mungkin waktu hadir
Terima
lagi
kasih.
Makoto.
Semuanya.
Oke. Oke,
Undang lagi
terima
minggu
kasih.
depan,
Oke,
Je.
terima kasih.
Dengan pembicara Dr.
Ya,
April.
minggu
Yes,
depan.
Allah.
Thank you
Mas
very
April,
much.
salam
Minggu
buat
depan,
Makoto.
Dr. April
Terima
minggu depan.
kasih. Mbak
Insya
Ina.
Allah. Mohon
Oke, izin
pamit,
Liv, ya.
matur nuwun.
Sami-sami.
Assalamualaikum.
Bye.
Nuwun semuanya.
Bye. Terima kasih sekali.
Makoto, thank you.
Thank you.
Tuh.
Masih ada, Prof. Makoto.
Ya, kita boleh live ya? Belum.
Iya, Liv, boleh.
Sudah.
Gak ada yang larang kok.
Boleh.
Ya, makasih.
Makasih.
Makasih Rania
semuanya.
ya, makasih Rania.
Iya
Makasih.
dokter,
Saya kok gak diterima kasih sama
Oh
Bu
ya,
ini.
makasih.
Saya kok kira, wes, aku gak diterima
Ya,
kisi
ya,
sama Bu
ya.
Enteng.
Makasih, makasih.
Bu Sylvia, terima kasih Bu Sylvia.
Ya,
Bu Nunu,
ya,
terima kasih
Ya.
semua. Terima kasih Bu Sylvia.
Ya.
Fasirin, Bu Lucia. Terima kasih kehadirannya.
Ya.
Oh iya, bukan Dara, bukan Liv. Terima kasih.
Mohon izin untuk live.
Izin live, J. Terima kasih, mohon izin live.